Hello and welcome to our channel. This is Gauri and today we will be discussing about ribozymes, its classes, types, functions and applications. So let's get started. The term ribozyme is used for RNA that can act as an enzyme. Ribozymes are mainly found in selected viruses, bacteria, plant organelles and lower eukaryotes. The functional part of the ribosome, the molecular machine that translates RNA into proteins, is fundamentally a ribozyme. A ribozyme, also called RNA enzyme, is an RNA molecule which catalyzes a chemical reaction. In 1967, Carl Woese, Francis Crick, and Leslie Orgel were the first to suggest that RNA could act as a catalyst based upon findings that it can form complex secondary structures. Ribozymes are divided into natural and artificial ribozymes. Natural ribozyme is further divided into large and small ribozymes. Group 1 and Group 2 introns are large ribozymes, while hammerhead and hairpin are included in small ribozymes. Artificial ribozymes are synthesized in the laboratory based on the dual nature of RNAs as a catalyst and an informational polymer. Some known classes of ribozymes include RNase-P, group 1 and group 2 introns, hairpin ribozyme, hammerhead ribozyme, hepatitis delta virus ribozyme, and tetrahymena ribozyme. Now let us understand the biochemical reactions catalyzed by ribozymes. Many natural ribozymes catalyze either their own cleavage or the cleavage of other RNAs, but they have also been found to catalyze the aminotransferase activity of the ribosome. Ribozymes that are known today catalyze two types of biochemical reactions. First, cleavage of phosphodiester bonds, that is group 1 and group 2 introns, and second, synthesis of peptide bonds, that is peptidyl transferase. Last are the applications of ribozymes. The ability of ribozymes to inhibit protein synthesis by reducing the level of specific mRNA in the cell is the basis for the therapeutic potential of these molecules. To date, most of the work concerning the use of ribozymes targeted against viral proteins has been carried out on HIV. Different portions of the viral genome have been chosen as target sites. Many examples concerning the use of ribozymes targeted against cellular genes have been reported. Some of them are closely related to possible clinical applications as the ribozymes used were targeted against genes predisposing to malignancy. Ribozymes also present a promising tool in the field of genetic diseases. In particular, some connective tissue disorders can be considered good candidates for a gene therapy protocol based on the use of ribozymes. So that is it for today. I hope you enjoyed watching the video. Thank you.